Okay, good morning again, my class. How are you today? For now, we are still discussing about regulation of glucose. We are now going to discuss on glucose as a source of energy. Here, as you are seeing, and we are observing that almost 80% of carbohydrates in our food are converted to glucose during digestion in the alimentary canals. Fructose and galactose is the other main products of carbohydrates digestion. This means what? When saying how the food get converted, we are taking the food at 100%, but 80% of those carbohydrates in our food are converted into glucose during the digestion in the alimentary canals. The alimentary canals, you know, from the mouth, esophagus, stomach, then in the intestinal canals. There, the food get converted. We see those fructose and galactose known as the isomers of glucose. Those are the isomers. They have the similar formulas, but different functional group. The similar functional group also, but different structure chain. Yes, different structures. Then now we are seeing after absorption from the alimentary tract, fructose and galactose are converted into glucose in the liver, which means their structure get converted and comes to be like the one of glucose. Now, and therefore, glucose constitute more than 95% of all the carbohydrates saturating in the blood. 95% of the carbohydrates saturating in the blood made of glucose, which means what? Here it is easier for glucose to be shared with the body parts during diffusion, where it passes from the alimentary canal to the body parts by sharing with the process of diffusion. We know there it is the reason why it is in the great amount of the body. Body cells require glucose continuously for its various metabolic activities. Yes, this is known and understandable. It is necessary for the body parts to get glucose time and time, because even the metabolic activities can't stop. When the metabolic activities get stopped, it means that the body the organism functions also get stopped, and an organism get die. Now we are seeing, when saying metabolic activities, we remember it is the one said to be catabolism and anabolism. These cells directly absorbed glucose from the blood, where the blood exchanging the glucose and getting the other ones from the body parts, where it shares glucose. The glucose is there to be combined, and after being combined, the was got energy and then the was get removed in the cell, and the energy updating the cell or the parts activities. Once inside the cell, glucose combines with the glucose combines with phosphate moiety, or a part of phosphate to form what? To form glucose 6-phosphate with the help of enzyme glucokinase. Here we know the glucokinase are in the liver, and this conversion is taken in the liver, where the glucose combines with the phosphate moiety to form glucose 6-phosphate with the help of those enzymes glucokinase. And in the other parts, we are finding the exokinase in the most of the other cells. When they are happening a conversion of the glucose, here in the other parts, there will be an intervention of enzyme called exogenous. Now this phosphorylation reaction is irreversible and helps to retain the glucose inside the cell. Understandable here, please? You see, the phosphorylation reaction is irreversible. It is reversible for what? When it is reversible, maybe the other glucose converted maybe get back. By back, you see there must be the other problem. Maybe once the glucose may be got in excess or in insufficient. So it is irreversible for retaining the glucose inside the body. Now, however, in the liver cells, liver cells, reniotubular epithelial cells, and intestinal epithelial cells. The enzymes, an enzyme glucokinase phosphatase converts the glucose 6-phosphate back to glucose. You see what we have said there? The glucose, by the phosphorylation process, we have seen how the glucose have been converted into glucose 6-phosphate by the enzyme called the glucokinase, where glucose combine with the phosphate moiety to form glucose 6-phosphate. Now, when in the liver cells, reniotubular epithelial cells, and intestinal epithelial cells, the enzyme called the glucose phosphatase helps to convert glucose 6-phosphate back to glucose. This happens when glucose seems as insufficient or when it is needed. Now, you see the process how it is. We find the glucose and the glucose react with this energy, glucose and this energy, in the presence of glucokinase and the exogenous in those parts, as said, glucose 6-phosphate produce glucose 6-phosphate and the ADP. You see the other energy, ATP, that's using this term. It got reduced. We are seeing ADP. It was triphosphate, now diphosphate. You see where the energy has been used. Now, the other back, when the other cells, in the liver cells, reniotubular epithelial cells, and the intestinal epithelial cells, we are seeing how glucose 6-phosphate get converted also into glucose with the other energy also. You see, with the help of the glucose phosphatase, those taken in the liver, in the kidney, and intestines, where we observe those cells. Here, the information is saying the complete oxidation of the one molecule of glucose. You see, one glucose molecule, when completely oxidized, producing carbon dioxide and water inside the cell, and produces many energies. The energy produced, as it is at 38 ATP molecules, where those 38 ATP molecules comes from different parts. One can say the parts where those comes from. You see, the parts where comes from, we have two from glucolysis process. The other two from Krebs cycle process, and the 34 from oxidative phosphorylation. One can ask him or herself what's glucolysis, what Krebs cycle, and what oxidative phosphorylation mean. When saying oxidative phosphorylation, in oxidative phosphorylation, one has to know first what phosphorylation is. What phosphorylation is. Here, phosphorylation. The phosphorylation process phosphorylation is the addition of phosphate group, or addition of a phosphoryl or phosphate group to a molecule. Addition of a phosphate group to a molecule. In a biological system, this reaction is vital for the cell, for the cellular storage and transfer of free energy using energy carrier molecules. Phosphorylation is the addition of a phosphate group or phosphoryl group to a molecule, and we have seen the other information where that's in the biology, that reaction is vital in the cellular storage and transfer of the free energy using energy carrier molecule. We are having also, one can ask now, oxidative phosphorylation means what? Oxidative phosphorylation is a cellular process that harnesses the reduction of oxygen to generate high energy phosphate bonds in the form of adenosine triphosphate. Understood? As another one also can asking him or herself what Krebs cycle is. The Krebs cycle is also known as the citric acid cycle. The Krebs cycle is a chain of reaction occurring in the mitochondria through which almost all living cells produce energy in aerobic respiration. Understood? It uses oxygen and give out water and carbon dioxide as products. Here it's understandable, I think. We shall see. Another one also can ask him or herself what glycolysis means. The glycolysis means what? Glycolysis is the process in which glucose is broken down to produce energy. You see, it produces two molecules of pyruvate. This will be seen. ATP, NADH, and water. The process takes place in the cytoplasm of the cell and does not require oxygen. It occurs in both aerobic and anaerobic organisms. Understood, please? If you ask yourselves what aerobic and anaerobic organisms means, aerobic organisms are those organisms that are able to survive, to respire in the presence of oxygen, or to make a gaseous exchange in presence of oxygen, while anaerobic ones are those that are able to survive in absence of oxygen. They do not use oxygen as a gaseous exchange material. Understandable, please? Yeah, let's generating the questions and me generating the answers.